So today, we're going to unpack a really fascinating discovery. It pulls back the curtain on one of cancer's most cunning survival tricks. And you know what? It's not a direct attack. It's a secret war of starvation, and it's waged against our own immune system. Let's dive in and see how this invisible battle actually plays out. Right, this is the big one. It's the question that has really been bugging researchers for decades. I mean, we have this unbelievably sophisticated defense system in our bodies, and it's designed to hunt down and eliminate threats just like cancer. And yet, tumors still find a way to grow and spread. To figure out why, we've got to look at what's happening to our immune soldiers right there on the front lines. Okay, so let me introduce you to the CD8 plus T cell. Think of this cell as an elite, cancer-killing soldier in our body's immune army. When it's healthy, it is an incredibly effective assassin, patrolling our body, taking out threats. But when these exact same T cells enter a tumor, something goes terribly wrong. They become what scientists call exhausted. They're still there, but they've completely lost their ability to fight. They've effectively become prisoners of war. So let's zoom in on the scene of the crime, the tumor microenvironment. Now, this isn't just the tumor cells themselves, but the whole local ecosystem buzzing around it. And let me tell you, it is an incredibly hostile place for our T cells. All the evidence is pointing to the idea that the T cells aren't being beaten by a direct assault, but by something way more subtle. They're being starved of critical supplies. And that brings us to the crucial nutrient at the very heart of this story, methionine. It's an essential amino acid, which just means our body can't make it on its own. We have to get it from our food. It's a fundamental building block for pretty much everything a cell needs to do, and as it turns out, it is especially critical for our immune cells. To really pinpoint the problem, researchers ran this brilliant experiment. They took T cells, and one by one, they systematically deprived them of different amino acids. And the result was, honestly, it was startling. I mean, sure, removing other amino acids had an effect, but taking away methionine, it was absolutely catastrophic for the T cells. It caused the most dramatic cell death and dysfunction by a long shot. Now, to really get the scale of this battle, we need a baseline. In your healthy blood, the concentration of methionine is floating around 30 micromolar. That's just a unit of measurement for this tiny but vital amount of fuel. Try to remember that number, because that's the total supply that our T cells and the cancer cells are fighting over. And here is the cruel twist. In this low-resource battlefield, our T cells have a huge vulnerability that cancer cells just don't. They are far, far more sensitive to a lack of methionine. They just need more of it to survive and do their job, which, well, it gives the tumor a massive strategic advantage. And here you can see the result of that vulnerability in action. When you put T cells and tumor cells together in this environment, the tumor cells act like greedy, greedy hoarders. They just gobble up the vast majority of the available methionine, which effectively starves the very T cells that are trying to kill them. This, right here, is cancer's secret weapon. But how? How does a lack of one little nutrient actually shut down a powerful T cell? Well, the answer is in this amazing field called epigenetics. The easiest way to think about it is this. Imagine your DNA is a giant cookbook with thousands of recipes, or genes. Epigenetics is the system of sticky notes and bookmarks that tells the cell which recipes to use right now and which ones to ignore. It's the control panel. So this theft triggers a devastating domino effect inside the T cell. First, the tumor steals all the methionine. Without it, the T cell can't make a molecule called SAM. That's basically the ink for all those epigenetic sticky notes. So the second domino falls. A key on mark gets erased from the cookbook. This flips a critical survival gene called STAT5 into the off position. And then the final domino. With its main survival program turned off, the T cell just shuts down and dies. A complete system failure, all because one nutrient was stolen. Okay, so that leads us to the next obvious question, right? It's one thing to know that tumor cells are hoarding methionine, but how are they physically doing it? What's the mechanism that lets them be so much better at soaking it all up than our own T cell? Well, the culprit has a name. It's a protein called SLC43A2. It's basically a transporter that sits on the outside of the cell and just pulls methionine in. Now, T cells, they barely have any of these. It's like a tiny little mail slot on a door. But the tumor cells, they are absolutely covered in them. It's not a mail slot, it's a massive industrial vacuum cleaner, just sucking up all the methionine before the T cells even get a chance. And look, this isn't just a theory. The scientists who discovered this laid it out plain and simple in their 2020 paper in the journal Nature. They said, quote, tumor cells outcompete T cells for methionine via SLC43A2. It's this specific tool that the tumor uses to win the resource war. Case closed. So once they identified the problem and the mechanism, the researchers got to the most exciting part, trying to fix it. They came up with two really clever strategies to try and level the playing field for our poor, exhausted T cells. All right, the first strategy was pretty simple and direct. If the T cells are starving, well, let's just give them more food. So they injected extra methionine right into the tumors in mice. And the result? The T cells got revitalized, they went back to work, and tumor growth slowed way down. It worked. Now, the second strategy was a little more targeted. Instead of just adding more fuel to the fire, they decided to block the tumor's vacuum cleaner. By chemically blocking that SLC43A2 transporter, they stopped the tumor cells from hoarding all the methionine, and that left enough for the T cells to get their share. And guess what? Once again, tumor growth slowed down. But here is where it gets really promising. They took their new strategy, blocking the transporter, and they paired it with an existing immunotherapy, a checkpoint inhibitor. Now, on their own, both treatments had, a modest effect. But when they put them together, the result wasn't just additive. It was synergistic. It's like 1 + 1 didn't equal 2, it equaled 10. They created this supercharged anti-cancer effect that was way more powerful than either treatment alone. So let's just recap this incredible journey of discovery. We started with the problem. T cells get exhausted in tumors, and we didn't totally know why. Now we understand the exact mechanism. It's a metabolic war for methionine, and the tumor wins by using its SLC43A2 transporter. And this points to a really clear future opportunity. We can target this pathway to make the cancer treatments we already have work even better for more people. And that leaves us with this really profound final thought. For decades, our main approach to cancer has been to poison it, to cut it out, to radiate it, all-out attack. But what if a key part of the solution is less about attacking the cancer and more about just nourishing and supporting our own body's natural defenses? What if one of the best ways to fight cancer is simply to make sure our immune system gets a proper meal?